Hi, I'm Dr. Danielle Brandman from the University of California, San Francisco, and I'm here today speaking with Dr. Neil Theise from New York University. Dr. Theise, thank you so much for speaking with me today about a paper that you co-authored with Dr. Ryan Gill, also from UCSF, about the microanatomy of the liver. Now, first, I was hoping that you could tell me a little bit about the microanatomy and the individuals in which you included in this paper.
Sure. What we chose to focus on for the paper is that there are three structures of the microscopic anatomy of the liver that have names. There's Rappaport's acinus, which describes the functional unit of the liver at the very microscopic level. There's something called the space of Moll, which sort of all liver people have heard of and no one knows what it is. And the Canal of Herring, which had been known about for a long time, but it turned out not quite in the way it actually looks in reality. And we focused on these in part because each of those investigators, spanning the course of about 50 years between the mid-19th century to the very early 20th century, were applying new techniques at the time to looking at microanatomy, things like injecting colored dyes into livers, for example. And in doing so, they found structures that had not been seen before. And in significant measure, two of those in particular, the Canal of Herring and the space of Moll, have been subsequently revised. And those revisions in the anatomy have led to lots of important discoveries. And those revisions happened because new techniques had become available that hadn't been used before. So that was kind of the theme, how when you have a new way of looking at anything, but in particular, the microscopic anatomy of any tissue, you find things you didn't see before, and you can begin to explain things you couldn't explain before. So that's sort of the overarching theme of the history.
Got it. No, that's very interesting for sure. Now, I'm wondering, are there particular diseases within hepatology that are really benefiting from these developments?
So the Canal of Herring change in anatomy, that change was my work actually, and it came from looking at three-dimensional reconstructions of these tiny, the Canal of Herring is the juncture between the hepatocyte canalicular system into the biliary tree, so how bile gets from the hepatocytes into the bile ducts. And Herring thought it was only this long, and it turned out to be about this long. What the implications of that were when we looked at how it changed in disease is that that's probably the primary hepatobiliary stem cell niche for the liver. And our first paper was in humans, and it was that paper that kind of landmarked the idea that, okay, livers have stem cells. What their use is, how important they are, that's still debated. But do livers and do human livers have stem cells? Yes, and some of them reside in the Canal of Herring. So that had huge import. The more recent revision, the space of Moll, he injected colored dye into the hepatic vein and into the portal vein and the hepatic artery, and saw that both of those entered spaces in and around the portal tract where they mixed. And this is called the space of Moll. But when you look at the portal tract, it's really hard to see this space. That's because he actually looked at cat livers where it's more prominent. It wasn't human livers. What we saw in human livers is the dense connective tissue of the portal tract looks like it has little cracks in it sometimes, but that's just because collagen is stiff. It turned out that last year, using in vivo microscopy, we saw that the structures of the biliary tree, the connective tissue layer isn't dense connective tissue in living tissue. It's these open fluid-filled spaces supported by these collagen bundles. And it turned out this is a feature not only of the biliary tree, but the entire portal tract, and in fact, all connective tissue in the body. So this became the heart of this redefinition of the interstitium as a new organ system last year, which made a lot of news. But when you look back in the literature for people who had recognized this fact, probably Moll is the first one in recorded history, and he saw it in the portal tracts because he was doing these injection studies. And so the space of Moll is the portal interstitium, we would now call it. The implications of that We've got evidence to suggest it's a major route of cancer spread that hadn't been recognized before. It probably relates to regulation of inflammatory processes, of aging, et cetera. So probably protein implications for that.
Now, are there techniques or other methods that are being developed at this point to really help us better understand the structure and/or function of the liver?
Yeah, all the time. But it's not like there's a bunch of people who are sitting around thinking, how can we look at the liver better? These things happen because people interested in techniques of observation do things, and then people like me bump into them, usually by accident, and go, oh, I wonder if we looked at the liver like this, what would we find? So with the Canal of Herring, it was simply, it wasn't so much a new technique as just thinking, what do these things look like in 3 dimensions? So all we did was serially section tissue and print the photographs and then trace things down in 3 dimensions. The in vivo microscopy that allowed us to see the living spaces and the living tissue, that came about through the endoscopists. And at the time, I was actually working with David Carlock and Petros Benius at Beth Israel Hospital in New York. They're endoscopists, and they had this new PCLE scope, confocal laser endomicroscopy, and they saw something in the wall of the human bile ducts they were looking at. It is a clinically used device that looked like these spaces that had not been described. And I'm their pathologist, so they said, what's going on in this bile duct? And I said, I don't know. And we started to explore it. So that came out of completely left field. And finding it in the bile duct is a kind of odd place to suddenly find something that redefines human anatomy. But, you know, the liver's important, so.
Some would argue the most important organ in the body. At least I say that all the time.
I do too. Seat of the soul, according to the Babylonians, more history. So, yeah.
Now, what do you think the next frontier is of liver histology?
I have no idea. This is the thing, when we came up with the interstitium business, a very common question was, well, what's the next thing we haven't discovered? Well, if I knew that, we would have discovered it. I think what we can say with certainty is that there are people devising all sorts of interesting techniques to look at tissues in 3 dimensions, in 4 dimensions over time, new techniques of staining, new techniques of visualization, new ways of integrating data like that. And when someone in the liver world decides, oh, that technique could be interesting to look at in the liver, we will find extraordinary things we haven't seen before. And I'm kind of hoping I live long enough to see someone come along and see the things that we've seen and then tell me how they need to be revised, because that's going to be really exciting.
Yeah. Well, it sounds quite fascinating. It sounds like we've learned so much about the liver already, but there's still so much more we need to learn.
We know this much.
Yeah. Well, thank you very much for speaking with me here today and sharing your expertise and your work.
Thank you very much.